Hello, everyone. This is Pocket Size Bio with Dr. Casco. In this video, we will continue Chapter 1 on the molecular biology of the cell by Bruce Alberts, and we'll continue with this little sub-module that I've made, which is genome diversification and the tree of life, right? Each little module contains a little introductory paragraph that talks about what the whole module is about. So I've embedded this into the notes for you to get the whole scope of the book. All right, so let's get to it. Okay, so in this module, right, genome diversification and the tree of life. So we're talking about how organisms have branched out into the different domains, basically Eukarya, Archaea, and Bacteria, and how different domains have these defining features, right? But at the same time, we're still kind of related and unified by certain key characteristics, right? So, of course, as I've been mentioning, living organisms were based on DNA and then RNA to produce proteins have had over the whole entire course of life, um, tremendous success and thriving, right? And life through billions of years have proliferated, as we know, right? You see life basically all throughout the world, right? From the the deepest areas of the ocean to the highest mountains within Earth's crust, right? So life has expanded everywhere, right? And all this idea of life and biological activity in itself shapes the earth, right, through biogeochemical processes, right, and even geological formations like coal. We're all familiar with how coal and oil and this whole concept of crude oil comes from, right? But even limestone and marble, right, have some impact, direct or indirectly, made from organisms, right? So it's quite spectacular how life has, one, expanded throughout the world, and two, influenced, right, the land formations, right, or geochemical formations, right? So the scope of this little module, right, is living things inhabit diverse environments beyond what we commonly think about, when we think about the world, right? So, uh, yeah, beyond the familiar realms of land, water, and water, right? Of course, all encompassing with the energy of the sunlight, right? But there are some organisms that have been able to thrive in incredibly extreme environments. We're talking deep oceans, right? Deep ocean trenches, deep thermal vents, hydrothermal vents, right? Incredible pressure down in those areas, incredible temperatures, right? Absolutely no oxygen, volcanic mud, and incredibly cold areas, right? Antarctic pools, even within the crust and the soil and rock, right, of Earth. So, a wide variety of um, environments, right? So, but the thing is that the majority of these organisms that we think about, right, and um, are able to thrive in this area is actually microscopic, right, and fairly difficult to even observe due to the area, right, due to the environmental conditions. So the ones that primarily thrive everywhere is, the microbes, right, the microscopic organisms, right? Um, by definition, they're microscopic, so they're unseen, but they have proliferated to a wide variety of um, different species, right? Incredibly numerous on Earth, right? And it's only until relatively recently that we can say, with modern molecular analytical tools, that we can actually get a scope of how diverse this microscopic world is, really, and how their places in the whole scheme of life's diversity, right? So by by having genetic information, right, and really accurate DNA sequencing and relatively quickly, right, um, we can get a kind of overarching visualization of what life fits in a general scheme of things, right? So, um, yeah, having genetic information in DNA sequences provides a universal language for our organisms that enable us via the sequencing to characterize organisms, to catalog organisms by features, right, similarities, and compare organisms from each other, right, and how we could organize life, right? So by comparing, DNA sequences, you basically it basically helps, right, specify each organism's place in what we call a tree of life, right? And how they relate to each other and things like that, right? So the first little subtopic that we're gonna go into is basically the tree of life, and basically it encompasses three domains: the eukaryotes, bacteria, and archaea, right? So this deals a lot with classification, right? And classification basically enabling the organization of different animals, different organisms, not just animals, right? But all life. And in the past, it was traditionally done just by outward appearances, right? Physical features. These two organisms look alike, so we're going to put them in this or in this particular group. They have four limbs and they walk on four limbs, etc., etc. So, um, in this way, we kind of started getting a sense of organization, right? Of classification. So the more features you had that were similar, the more of close family resemblances, were interpreted in terms of these different organisms and in terms of evolution with common ancestors, right? Common ancestors referring by the analysis of fossil records, right? Um, that would give a sense of current species toward the past, right? Now, while that did help in a sense, there were definitely challenges with that idea of classification because, one, what how would you classify organisms that would be incredibly different, you know, and um, you don't see any form of similarity, right? Uh, so in that sense, organization and classification will start to get difficult, will become difficult, right? And even when you're looking down at the microscopic level, single-celled organisms, prokaryotes, right? Bacteria and archaea, where just by shape alone is incredibly, it's impossible to really get a sense of different species and furthermore to even classify, right? Until you use molecular sequencing, DNA sequencing, right? And um so these microscopic organisms, like again, like I mentioned before, like bacteria, they do present further classification challenges, right? So, and on top of that, the idea of just being able to distinguish them, a lot of bacteria, a lot of microbes are quite difficult to culture in the laboratory, right? They require very refined environmental conditions to actually thrive in there, right? In a laboratory setting. So it's actually not a lot of organisms that could be cultured um, effectively in the lab, right? So, but as we collect different samples, we can, again, DNA sequence, and by DNA sequencing the bacteria in particular, microbes in particular, you can start to get a sense of microbial biodiversity, especially those that you sample in extreme environments, right? So, um, in that sense, you can get a whole relationship in much more detail, right? So that idea of sequencing ultimately brings down the idea of genome analysis and evolutionary relationships, right? So again, genomic analysis can help provide a direct and incredibly powerful way, right, to determine evolutionary relationships, right? And by analyzing the genome and by obtaining precise DNA sequences, comprehensive DNA sequences of genomes, right, you can really define an organism, right, pretty well and compare with other organisms, right? So by having that, you can get a sense of how related certain organisms are to a very accurate degree, right? And depending on the number of differences within a DNA sequence, you basically can make a kind of evolutionary distance quite quantitatively. And one of the molecular rulers that scientists use is basically ribosomal RNA sequences, because it's um, one of the sequences that is used so much and conserved throughout species, basically essentially being used for protein manufacturing, right? That should be something that should be found, that can be found um, in life, right? In organisms. So an interesting little diagram that was found, that is in the textbook is, this one that you see here, which is a global tree of life based on genome comparisons, right? That show the three major divisions, right? Or the domains that we think about, right? And the book mentions, which is incredibly interesting. What we think about life, right, is a very small portion of the whole tree of life. So if you take a look here, the vast majority of variety, right, of living organism is actually bacteria, right? You have these different groups of bacteria. And then you have Archaea that you don't have as much variety. And even so with Eukaryotes, right, is just this little branch of biodiversity when you compare to the whole scheme of life. Very, very interesting. And yeah, based on molecular sequencing, right? So it's quite interesting how diverse life is, but we don't really see it because it's in this um you know microscopic, you know um um form, that is, it's difficult really to see in a day-to-day basis, right? So but nevertheless, you have this concept, right? A variety of life creates this whole um an evolutionary tree, right? So having this ribosomal RNA sequences, of course, analyzing DNA sequences, we essentially get three major divisions of the living world, right? Which we probably remember from high school, which are the eukaryotes, bacteria, and archaea, right? And then these three, of course, represent distinct evolutionary lineages. All right. So focusing a little bit about eukaryotes, right? They make up the domain of life that is actually the most familiar to us. What we think about as life, we're pretty much focusing on eukaryotes, right? And this gets the features about eukaryotes goes in a lot more detail in the following part, and of course throughout the entire book. But just to kind of summarize some of the features that belong to eukaryotic cells, right? You have organisms that have an enclosed DNA is enclosed within a membrane-bound nucleus, right? They're generally larger in size compared to bacteria and archaea. Um, they contain membrane-bound organelles, as we're going to see in the second part. In the third part, mitochondria, the ER, Golgi apparatus, and many others, right? Organelles, and definitely the genome tends to be a lot larger, more complex, with many more genes, right? spanning tens and thousands of genes and more, right? So having this level of complexity means that you can diversify the different types of organisms, right? You include the plants, animals, fungi, and protista, right? The protists. And within these groups, you can have single-celled organisms, or you can have multicellular eukaryotes, you know, um, that further kind of enhance that level of um, variability, right? Which ultimately extends to what we think about as life, right? Demonstrating diverse life strategies and structures. And the figures you see here are some examples of how different shapes and sizes of certain cells when we now focus in prokaryotes, right? Let me zoom out so you can see better here. We start to see the most diverse group. So we're moving from eukaryotes, talking about prokaryotes now, and bacteria, right? And I'm going to go to these panels in a bit, but just to give you some sense of characteristics about bacteria. So they have immense genetic and ecological diversity, and they're characterized by being very small in size, always smaller than eukaryotic cells, right? Let me just expand this so we can see better, right? And bacterial cells tend to have or have their genomic DNA as in a circular single chromosome, right? And generally in a particular area, which is referred to as the nucleoid region, but you can see the DNA, right, is condensed in this central area. Doesn't necessarily have to be in the center, but it's around this particular region here, right, where you have circular a circular chromosome, and aside from a circular chromosome, they have little circular DNA, much smaller in sequence length, that are referred to as plasmids. They contain additional information. They are used a lot in biotechnology, that we're going to see later on. But they can help transfer that, to other bacteria and whatnot, to, for example, provide resistance to antibiotics, for example, right? So aside from the structure, they inhabit a wide variety of environments, from soil environments to aquatic extreme environments like hot springs and deep sea vents, right? And they're very, very important when discussing about nutrients, biochemical cycling of material, breaking, for example, nitrogen, right? Um, Decomposition to break down organisms, and even create symbiotic relationships with other organisms, right? So aside from that, uh, um, many bacteria actually are quite pathogenic to plants, animals, including humans, right? Where they can cause some devastating diseases um, that we know about, right? So just to show you some panels, again, bacteria can become are very diverse in their genetics. They do have some morphological forms that you see here, right? Spherical forms that could be in chains or by themselves. Basically, the um, Streptococcus, right? Bacilli form, or the coccus form, and then the bacilli form, basically as a rod, or a sphere coccus form, right? Um, and then so forth. We can also have little spiral forms, right? That you can see here, all right? Like the spiroplasma here. Okay. So you could have not an incredible diversity in terms of form, right? Spherical or bacilli or spiral, right? And their general size is much, much smaller, right? Um, Compared to an eukaryote, right? Again, like I mentioned before, there's some specialized type of bacteria that can photosynthesize and even, help in the formation of oxygen, right? So, continuing, we have Archaea, right? Very, very mysterious domain of life. And they do share some characteristics with bacteria and eukaryotes, and but they have their own unique molecular characteristics. Um, they seem to have a little bit more similarities with eukaryotes than bacteria, actually, but they're often characterized and kind of known for living in extreme environments, right? Incredibly extreme environments, right? So you have hot springs having high temperatures or incredibly salty environments, right? Even in conditions that are one of the most severe, which is the deep sea hydrothermal vents, right? As I mentioned before, right? Their cellular composition in terms of cell wall and membranes are quite unique that we're going to see later on. And um, as I mentioned before, right, some genomic studies and comparisons kind of show that archaea are much closer to eukaryotes than what has previously been known, as opposed to being closer to bacteria, right? So again, archaea, they being prokaryotes, they do play essential roles in the biochemical cycling of certain um, uh, material, right, throughout the world. Geochemical processes, the nitrogen cycle, methane production, right, and so forth. So um, definitely studying them can give a sense of how they've helped in formation of the earth, and um, some sense on how the early environment as was, right? So focusing more on the general scheme of things now, organisms occupy most of our planet, right? So um, in terms of, it's quite interesting how the book kind of plots in terms of the biomass, right? Uh, Gigatons, right? If we were to observe and take a look at all the habitats on Earth, right, from terrestrial and aquatic and the extreme environments, um, approximately there are 550 gigatons, right? And sorry about this little the little annotation. I couldn't put the little exponential indicator here. 10 to the 15 grams of carbon, right, forms the total biomass on Earth, right? And among that, most is from plants, right? So if you see this little chart here, plants provide, if I can say, out of that 550 gigatons, 450 gigatons of carbon come from plants, right? And then you could see, of course, the different figures here. I won't go into the specific numbers here, as you can see. But what is interesting is that animals, what we think of as the most diverse and um, overrunning the world, animals only provide a 2 gigaton of carbon of biomass toward the total on Earth. So that's quite interesting, right? Um, in terms of the total terrestrial biomass, it is 100 times greater than the biomass coming from the ocean. But the although most of the animal's mass, right, is actually found in the oceans, right? And in terms of humans, humans have a biomass that is 10 times greater than all measurable wild animals altogether. So it is quite interesting in terms of human population in our place, in terms of biomass compared to all living organisms, right? Okay. So aside from that, as basically you can see life all throughout different places around the world, right? Organisms have evolved, right? Have adapted, have developed features to thrive in very specific ecological niches, right? And based on environmental pressures, temperature, moisture, nutrient availability, over time, organisms are able to adapt to thrive in these particular situations, right? Um, and even microorganisms, right? Bacteria and archaea, they're able to inhabit almost every ecosystem, if I can say, on Earth, right? Even those that were thought inhospitable, right? They thrive in there. Of course, one aspect that does need to be mentioned in terms of cell biology and just biology in general is human impact, right? Human activities have influenced a great um, change in the distribution and abundance of various species throughout the world, right? Habitat destruction, pollution, climate change, all um, coming from human activity has forced many species to kind of get displaced or sometimes even whole ecosystems broken down, right? Um, so by understanding, habitat diversity and those organisms that live in these places, right? It's important because you can get some sense of conservation efforts, right? And ecosystem management. What are some do's and don'ts? What are some more effective methods to maintain those species that are there from basically extinction, right? So, um, with that aspect, so how do organisms even thrive in there, right? How do we get these organisms to be able to survive in these particular environments, even those that we think that are inhabitable, right? Of course, life, you need free energy, right? You need some form of energy. So where do you get that? And definitely we go into detail in the later chapters, but um, yeah, different organisms employ different strategies, different methods to obtain energy. We are familiar with phototrophs, right? Phototrophic organisms, basically the photosynthesizers, right? Plants, algae, bacteria. They harness the free energy from sunlight via the process of photosynthesis. They're able to create organic compounds from inorganic components, right? Carbon dioxide, water, and so forth, right? Um, and essentially, life on Earth and on Earth's crust, even in the oceans, right, on land, ultimately depend on phototrophic organisms, right? But there's some organisms that, really don't necessarily need, you know, photosynthesis and even oxygen for that matter, right? To thrive, right? And this is an example of organisms, microbes that are not just microbes, but lithotrophic organisms that can extract energy from inorganic compounds, right? In this case, as you see in this little diagram here, in these hydrothermal vents that I'll get into in a second, right? But in general, organotrophs, right, highly reliant on phototrophic organisms or other little lithotrophic organisms that we see in these deep ocean vents, right, as a form of energy. So basically, consumers, right? So, in terms of organotrophs, that's what we commonly think about as consumers, right? You have animals, right? Eating other organisms and essentially how these consumers go on about throughout the ecosystem can highly influence the dynamics of an ecosystem or the sustainability of the ecosystem, species interaction. Um, if you probably studied in ecology, the different types of interactions, most commonly predator-prey relationships, right? But even biogeochemical cycles, right? What is interesting is that in this whole concept of consuming organisms to break down their food and produce organic compounds via cell respiration, right? You do require oxygen, right? Um, Aerobic conditions, right? But some organisms don't, it's not even necessary to have oxygen, right? Um, and the book puts some emphasis on this, on particular um, archaea, right? That are able to thrive in this incredibly unique and intense environment, right? Extracting um, chemicals from these hydrothermal vents, you have this situation where under no situation you have oxygen, but through a certain cycling of material, right? You have this deep ocean vents in Earth's crust at the bottom of the oceans where you have percolation of seawater, salts and minerals are coming down, heating up with hot rock layers, basalt, right? As this is occurring, you're getting a mixture of nutrients, of elements. Initially, at the base of these vents, it's incredibly hot, right? 350 degrees Celsius in this core. As differences in temperature, right, gives the differences in pressure, you're having an upwelling of material. And as it's going up, right, you're releasing gases, you're releasing material, and it's cooling down greatly. As this material's coming out, it's, slipping down these little the walls, okay, of these vents, right? Cool enough for certain bacteria to thrive in, right? Those upper areas. And as it's falling, getting cooler and cooler and cooler, you're even getting invertebrate communities that are living there that are absorbing material from the environment, that are even having symbiotic relationships with anaerobic bacteria to kind of process these inorganic compounds to be able to thrive together in this ecosystem. So it is quite fascinating how um, um, you can have a living environment in extreme conditions, right? And you have nutrient cycling even without the need of sunlight, right? This process of chemosynthesis, right? That we get into in the later chapters again. Okay. So some examples that you see here. Again, you get much more complex organisms, multicellular animals, tube worms, mussels, um, and so forth, right? That are able to thrive in this environment. Okay. So among these, right, you have some organisms, not just in the thermal vents, but going back into the general scope of life and in the world, um, key players, right, in the idea of cycling material. Um, First, it's necessary to be cycling material, especially carbon dioxide, especially nitrogen, right? Living organisms, life in general require, I'm going to say require nitrogen, right? Of course, carbon dioxide to basically produce, all these biomolecules. We need nitrogen and carbon. Of course, you study organic chemistry there, right? To as a basis, right? To produce these molecules, right? DNA, RNA, protein all require carbon, hydrogen, nitrogen, sulfur, oxygen, phosphorus, right? And we know that nitrogen is readily available um, in the air, but really we can't fix or we can't change nitrogen gas, N2 gas, in a form that we can assimilate, right? That we can absorb. So we need the help. Organisms, animals need help from nitrogen fixing bacteria that can convert atmospheric nitrogen into a form that is useful to us, that we can assimilate. Um, Ammonia, for example, right? Plants can absorb, other organisms can absorb ammonia and can change this into a form we could use, right? Into a structure as part of the DNA, for example, right? So whenever you change the form of nitrogen from one compound to another, that's called nitrogen fixation, right? You're fixing nitrogen in a way that we can use. And the same goes for carbon dioxide, right? Nitrogen and carbon dioxide are quite stable, incredibly stable, and require a lot of energy to change in their um, chemical form, right? So we need help from um, plants, algae, bacteria, right? Nitrogen fixing bacteria to be able to change these things, right, into a way that we can assimilate. Okay. So much so that many plants have had a symbiotic relationship with nitrogen fixing bacteria, and it enhances the overall fertility of the soil, And some plants like the pea plant even have little nodules that they can house these nitrogen fixers to be able to produce nitrogen for them, right? So incredibly important, right? To understand nitrogen cycles and carbon dioxide cycles because we use it all day, every day, right? So of course, it's definitely important for agriculture, right? Climate mitigation, ecosystem health. Measuring these factors is important so we can understand how healthy the ecosystem is. Okay. So, going on to genomes diversify over evolutionary time, right? Producing new types of organisms. This is incredibly interesting. And this has to do with genetics and mutations, right? Um, Evolutionary processes do promote or drive genetic diversity, right? Basically, sequencing differences, right? And adaptation for every species at the population level. And one of the main things we're going to see in terms of genetics is that mutations add genetic diversity, right, for a particular species and their sequence, right? And cell division, one of the major players during cell division, you can get some errors in DNA replication, right? That could add creating errors can add mutations. Now, mutations is by definition is a permanent change in the DNA. Now, a permanent change in the DNA sequence can be something positive, right? Have something beneficial for the organism, for the population. Could have, and I designate it as these numbers here, right? Could have a neutral change in the DNA sequence. You don't improve or um, make the sequence become deleterious, or it becomes something bad, right? And what's important is this idea of perpetual, right? Being able to pass down. If it's something beneficial, the organism will thrive better, the species will thrive better, and they will succeed in passing in generations that new mutation, that new protein being made through generations, right? That's the idea of being perpetual. Um, but if it's negative, most likely the organism will die, right? The mutation of the DNA, especially if it's a vital um, gene, will most likely not let the individual survive, right? So, um, all of that encompasses this idea of altering DNA, right? We're familiar with the idea of mutations, and a change in DNA will alter the protein, ultimately create a phenotypic difference, right? And what I like how the book mentions it's a cycle of trial and error, right? Trial and error, little change, little change. If it works, we're gonna keep it. If it doesn't work, we're gonna remove you out. It didn't help. Let's try again. So that's what it refers to, kind of as a trial and error, right? So you have a mutation, natural selection. If the nature selects that it's worthy, it continues, right? That ultimately drives organisms to evolve as a species, right? We'll get into that in a lot more detail in the later chapters. Um, so um, yeah, some sections of the genome can change more easily than others. This is quite interesting in the sense that when you have mutations in sections of the DNA that doesn't really code for a protein or isn't as vital of the protein, you're gonna have um, since the organism's not dying, you're gonna have a higher frequency of mutations in these particular areas, right? Whereas if you have essential genes, you're not going to have mutations in these areas as much because if you have a mutation on these sequences, most likely the organism or the cell will die, right? Meaning that these genes are highly conserved, right? Highly, highly conserved. And one of those, right, being ribosomal DNA, right? Um, so, um, throughout time, and these mutations have been happening over an incredibly long time, 3.5 billion years uh, plus, right? You've had much time to have many changes of DNA. But again, like I was mentioning, those essential genes most likely are going to be conserved among different organism groups, right? Um, and that's why ribosomal DNA, being fundamental to protein production, is quite conserved, right? Quite conserved. There are some changes, and you can see an example here, right? Where you have a sequence a little sequence section of ribosomal RNA, right? Between humans, a methanococcus, and E. coli, and they put humans here again just for easier in comparison. If you were to see a human and a methanococcus, you see there's a lot of nucleotide sequences that are conserved there, right? Basically, these little red lines are showing conservation, right? Areas that don't have that red line, basically, it's a difference, right? Um, right, and even amongst these different organism groups, E. coli with this uh, um, Archaea bacteria, right? Archaebacteria or Archaea, you see some conservation. Different cell types still containing similarities within a sequence of a conserved gene that is necessary for DNA expression, right? Or gene expression. So very interesting there. Okay. So new genes are generated from pre-existing genes, right? We know that the raw material for evolution is a DNA sequence, right? And as I mentioned before in the previous video, DNA sequences can't be self-assembled randomly, right? You essentially needed to have a template, right, or something that already exists for you to make these subtle changes, right? These minute changes over time, right? In that sense, no gene is entirely new, and the way that you would create new sequences is, or one of the major ways, by mutations, right? So that idea of innovation, right, can occur in the following ways. So one way could be intragenic mutations, right? And these are basically mutations that occur as errors during DNA replication and repair, right? We've mentioned the process of DNA replication very, very briefly, and among that, that process, when you're adding those free nucleotides, you can have some errors occurring there, right? So you have intragenic, if you see here in the little panel here, intragenic mutation, where you have a gene in this particular DNA little diagram, and then you can have a particular area in a specific place, right? Leading to some genetic innovation, right? One way could be gene duplications, right? A gene gets duplicated, and where you would essentially make a pair of identical genes, right, within a single cell. And throughout time, these two genes that are duplicates of each other, right, will could diverge, right, into something new. You can have DNA segment shuffling, right, where you have two or more genes, as you can see here, gene A and B, and you can have certain sections of one, segments of one gene pretty much transpose with the other, and they basically kind of recombine to create hybrid genes, right? And at different locations, right? That could lead to innovation, right? And also essentially horizontal DNA transfer, intracellular transfer, where you're getting a piece of DNA from one organism and a piece of DNA from another organism. Here, for example, organism A and organism B, and they can transfer, genetic material, right, from one organism to another, right? In this case, organism A that has these orange, yellow, and red, genes is passing um, a gene, right, or segment of DNA to another organism, right? Let's say a bacterial cell. Okay. So uh from whatever method, right, any change will leave a particular trace of that happening, right, within the genome, that newly established DNA sequence in the organism's genome, right? And that's something that scientists can analyze. Okay. Um, Gene duplications give rise to families of related genes within a single genome, right? So it's quite interesting, accidental gene duplications right, lead to the presence of identical gene copies within a single cell, right? And this does happen where it does occur where it does happen where you have duplications occurring in parts of the genomes, right? So you're getting these identical genes within a single cell, and over time, some of these duplicates, right, can change over time, slightly, majorly, right? So they can acquire new functions over time. So you have some similarities, but you have some form of uniqueness within these duplicates because it stems from this little um, uh, base gene, right? Therefore, since you have some form of similarities, you can refer these genes that have some degree of similarities as gene families, right? So gene families emerge as duplicated genes diverge in function, right? While retaining similar sequence, right? Sequence similarities, right? And here's an example in this panel here where you can see, let me just bring it here. This is an example, for example, sorry, um, of the genome of Bacillus subtilis, right, of this bacterium, consisting of 77 genes coding for varieties of a class of membrane transport protein called ABC transporters, right? Um, and among this group of transporters, you have a certain, quantity of genes that are fixed within a particular family, right? Function, and so you have several genes that belong to these particular families, right? Um, so, again, from that same concept of duplication and divergence, right? You can analyze certain gene families there. Okay. Um, Gene duplication and divergence, they contribute to genetic robustness, right? And by providing more duplicates, you have an opportunity for the organism to adapt to changing environments, right? Um, and we're going to see this in a lot more detail in the later chapters. But genes of two separate species, these are concepts you're going to see in a lot more detail. Genes of two separate species that derive from an ancestral cell gene, right? And in the LCA, or latest common ancestral cell, are called orthologs. So let's say you have an ancestral organism via speciation for whatever X reason, right? Um, Gives rise to a species A and species G, where you have this um, little subgenes of gene G, for example, gene sub A, gene sub B, right? In different species, but that they still stem from this original gene, right? They're called orthologs, right? And any related gene, continuing on to another related term, right? Related genes that have resulted from a gene duplication event, right, within a single genome and have diverged in their function, right, is referred to as a paralog. So you have an ancestral organism with that original gene, right? You have a duplication where you have that a duplicate, right, of the original gene, and you have these two genes that have basically diverged in their function. So you have G sub one, gene G sub two, that maybe their function has changed, right, greatly within that organism, within a later organism coming from an ancestral organism, is referred to as a paralog, right? Ultimately, anytime you relate to any um, a broader term between an ancestral organism passing down genes to more current species, right? It's in general a term called homologs, right? That we're going to see more in detail. Okay. One of the last topics of this little module here, the function of a gene can often be deduced by its nucleotide sequence or from its nucleotide sequence. Um, Definitely, nucleotide sequences, they provide clues as to what a gene will do, right? And even evolutionary history when we compare different species, right? And comparative genomics, right, enables researchers to identify genes across a diversity of organisms, right, via sequencing and um, wide variety of organism bioinformatics and relationships, right? So um, known functions, right, of homologous genes basically can help predict the functions of newly discovered genes. So it's quite interesting as new sequences are being discovered just by a kind of comparative analysis between what is known and databases that have been submitted, for example, in NCBI, you can get some form of functionality just because of preserved regions, for example, in a particular sequence, right? So deciphering gene function enhances our understanding of biological processes and organismal biology, right? So, lastly, for this little module, more than 200 gene families are common to all three domains of life, right? So, complete genome sequences, they do facilitate the identification of gene families, of shared gene families among all domains of life, basically all life, right? Eukaryotes, bacteria, and archaea. And it's very interesting to see that we have some form of common genetic heritage, right, among all living organisms, right? This does become a problem where we want to obtain some more specific insights, but some genes are completely lost throughout time, right? Some genes get incredibly changed over time, so it's a lot more difficult to put the pieces of the puzzle together, and um, due to horizontal gene transfer, it's a little bit more difficult to identify where did we have this ancestral gene really coming from when you're having lateral transfer between different species, right? And in a current time, right? So um, there are some challenges in order to really provide a kind of genetic lineage to fully understanding living organisms and their genetic history, right? So, um, as I mentioned before, some genes in the course of 2 to 3 billion years ago have changed so much that it's even difficult to identify where they actually came from or what their ancestral gene was coming from, right? But of course, despite, these difficulties, there are some efforts in identifying conserved genes families, right? That can give some sense of commonality between all living organisms, right? And evolutionary relationships, right? So it is known, right? Genetic analysis, there's around 264 ancient conserved families, right? It's the largest number of shared gene families, right? Interestingly, they involved in translation and amino acid metabolism, right? In transport within the cell. So that could give a sense also on the importance of these mechanisms, right? And here you can see a table, a more specific table, right, on the number of gene families that are classified in this sense by function that is common to all three domains of the living world, broken down into information processing, cellular processing, and signaling, metabolism, and some that have been sequenced and have a predicted biochemical function, but the specific role, right, has yet to be determined, right? Okay, so with that being said, we finish this particular module. There's one more module to go, plus an additional module that hasn't been in, I believe, in the previous versions of this book, which is model um, organisms, or an updated version of that. So I'm going to include that, not necessarily as part 4, but I'll add it as a part chapter 1.5, for example.